This tutorial describes how to perform any absorbance or colorimetric based assay from preferred cell systems. This readout or endpoint is incorporated into all of the HemoLIGHT, ImmunoLight, MSCLight and STEMLight assays. If you have followed the tutorial on measuring fluorescence, you will notice that there are many similarities in the absorbance readout protocol. All assays that incorporate an absorbance or colorimetric readout or endpoint use an absorbance plate reader set at 490 nanometers or between 450 to 540 nanometers. You can also use a reference wavelength of 630 to 700 nanometers. Assays that measure absorbance are the most common compared to fluorescence or luminescence assays. Absorbance assays are usually the most cost-effective, but are also not as sensitive as other readouts. Like bioluminescence and fluorescence assays, the absorbance assays from preferred cell systems are cell viability assays, but also quantitatively measure changes in cell proliferation ability. The absorbance readout correlates directly with fluorescence, bioluminescence, and even radioactive readouts such as tritiated thymidine to measure cell proliferation. Results from absorbance assays can be normalized by performing a cell dose response every time an assay is performed. This allows a better comparison between experiments. Now let's take a look at how an absorbance or colorimetric readout actually works. Live cells possess functioning mitochondria that are capable of chemically reducing molecules. This bioreduction process by cells serves as a viability marker. The assay kit contains a ready prepared reagent called MTS, or Owen's reagent. This is a tetrazolium compound that is also combined with an electron coupling reagent to form a stable solution that is added directly to cells in culture. The reagent is added to each well at 20 microliters. Once inside metabolically active cells, it is reduced by NADPH or NADH from dehydrogenase enzymes, producing a soluble yellow formazan compound that develops over a 1 to 4 hour period. The amount of absorbance at 490 nanometers indicates the number of live cells present. You can also use wavelengths between 450 and 540 nanometers and a reference wavelength of 630 nanometers to 700 nanometers so as the background is subtracted. The assay can be normalized by performing a cell dose response curve at the time the samples are measured. Compared to an MTT assay, which also incorporates the production of a formazan compound, no solubilization step is required. The MTS assay is a single reagent, single-step assay. All absorbance or colorimetric assay kits contain nearly everything you need to culture the cells and measure absorbance. Now, some assay kits can be used on numerous cell types, and in these cases, the investigator should use their own culture materials and protocols. However, in general, the assay kit contains a base medium. This can be used as a background, ready-to-use MTS reagent, clear, sterile 96 well plates, and sterile adhesive foils that can keep unused wells sterile. Now, there are some supplies and instruments that you do need to provide yourself. For example, cells, if you're going to normalize the assay, and sterile tubes to dilute the cells for the cell dose response. A single channel pipette and sterile tips, a reagent reservoir, an 8-channel pipette and tips, and a plate reader that can measure absorbance at 490 nanometers. Alternatively, you can measure absorbance between 450 to 540 nanometers. You can also use a reference wavelength of 630 to 700 nanometers. Now let's go to the actual protocol. For this tutorial, it is assumed that the cells have been cultured for a specific period of time in the clear, sterile 96-well plate provided with the assay kit, because in this tutorial only the procedure for processing the plate and measuring absorbance is described. Now make sure your absorbance plate reader can perform the measurement at 490 nanometers. Also make sure that the instrument is adjusted for the correct gain if necessary. First, you will need to remove the base medium and MTS reagent from the assay kit in the freezer and thaw them at 37 degrees Celsius in a water bath. When thawed, mix gently by inversion several times, making sure not to cause any bubbles. Remove the 96-well plate or culture plate from the incubator and let it come to room temperature. As you see, we are using only 6 columns of the 96-well plate. This plate will now be covered with the sterile adhesive foil that's included with the kit, so that the unused wells will remain sterile for later use. To do this, place the 96-well plate in a hood to keep the plate sterile. Remove the lid. Peel off the back of the foil and press the adhesive side onto the top of the plate firmly. With a sharp knife or scalpel, the foil will be cut, in this case, between the 6th and 7th column. The foil is then removed from the first 6 columns of the plate, leaving the remainder of the plate sterile. The base medium has already been added to the first column. This will be your background. However, if you are using a medium containing serum, this should be used in place of the base medium, since serum contains substances that might produce an absorbance signal. This background needs to be subtracted from the sample values. Now pour the MTS reagent into the reagent reservoir. Using a multi-channel pipette, dispense 20 microliters of the reagent into each well and mix gently by pipetting, making sure no bubbles are produced. Transfer the plate back to the 37 degrees Celsius incubator. The absorbance signal will start to develop after about 1 hour as a yellow color in the wells. The signal should reach a maximum after 4 hours. Usually a 3-hour incubation is sufficient. You can remove the plate at any time to measure the absorbance to determine the optimum time for the cells being assayed. Don't forget to subtract the background value from all of the sample values. You might also consider using a reference wavelength of 630 to 700 nanometers, also as a background, to take non-specific absorbance or cell debris into account. We'll now briefly consider normalization of the assay. Since the method determines the number of live viable cells, the assay can be normalized by performing a cell dose response curve in parallel with the samples being measured. Always include a 0-cell control. A typical cell dose response curve may include cell concentrations from 250 cells to 10,000 cells per well, each in 0.1 milliliters of total volume. The cell dose response will be fitted to a linear regression curve fit, and the absorbance units obtained for the sample can then be interpolated as live cell concentrations. If this procedure is performed every time samples are assessed, it will be possible to compare results well to well, plate to plate, and day to day. It should be pointed out that this is not the same as performing external standards and controls, as is the case with the ATP bioluminescence assays from preferred cell systems. This concludes the tutorial on how to perform an absorbance or colorimetric readout for assays from preferred cell systems. If you have any questions or comments, please contact us. We're here to help you. Thank you.